Dear Grade 12, I am Mrs. V. Ramlal. Welcome to the Junior Tucky's Winter School for Grade 12s. There will be a series of videos which will be live streamed in the next few weeks on Junior Tucky Online. Please use this material as revision and also look at all the Grade 12 content in Life Sciences on Junior Tucky Online. In this video, we will be looking at the November 2021 Paper 1. Right, let's get down to question 2. Now, the diagram below represents a type of neuron found in the human body. So if you look at this neuron, this is the neuron we're looking at. It says 2.1.1, identify the type of neuron shown. Now, you've already drawn this neuron and you should know that this neuron is a motor neuron. Now, 212. Use the letters A, B, C and give the correct sequence for the transmission of an impulse along neuron 1. Now, we already know that dendrites, these are the dendrites, take the impulse to the cell body, and the cell body is the place where the nucleus is. So the impulse will enter here. The axon, which is this long part here, labeled B, will take the impulse away, and that's the terminal end plates. So the impulse travels into C, then B, then A. And that's how you write your answer. Because they're asking you sequence, remember to put arrows. 213. Explain how the speed of transmission of impulses will differ for neuron 1 and 2. Now we can see already colored in yellow. That's the myelin sheet. Now we know neuron 1 has a myelin sheet and neuron 2 does not have a myelin sheet. Now we know the myelin sheet is there for speeding up the transmission of the impulse. So neuron 1 has a myelin sheet and the impulse will travel faster in neuron 1. Neuron 2 does not have a myelin sheet and the impulse will travel slower. Make sure you compare the two neurons. 214. Why a person will feel the stimulus but will not be able to respond if this type of neuron, the motor neuron, is damaged. So, the motor neuron is damaged. You can still feel the impulse because the sensory neuron is not damaged. So, the impulse from the sensory neuron will still be transmitted to the central nervous system. But the motor neuron is damaged, so the impulse will not be transmitted to the effector to respond to the stimulus. Right, that is question 2.1. I'd like to quickly revise the structure of the neurons. You are asked to learn the motor neuron and the sensory neuron. Now, let's look at the structures. This is the motor neuron and these outgrowths are the dendrites. The dendrites take the impulse to the cell body. Now, how do you know this is the cell body? Any place that has a nucleus, that's the cell body. So this whole part here, with this bracket, that's the cell body. Then, of course, that's the nucleus, and then the structure on the outside is the cell membrane. All of this makes up the cell body. So this whole part is the cell body. So dendrites take the impulse to the cell body. This long structure here is the axon. The axon takes the impulse away, A for away. Around the axon is the myelin sheet, and that's the sheet that's responsible for speeding up the transmission of the impulse and for electrical insulation. So there I have direction of the impulse. Dendrites to the cell body, axon away. So the impulse travels in this direction. This is a sensory neuron. There's the cell body again. The cell body is the area that has the nucleus. Now in this case, this is the, let me just look and see, there's your axon and this is your dendrite. So dendrite to the cell body, axon away. So the direction of the impulse goes this direction, which is from left to right. So this is the structure of this motor and sensory neurons. Now if you look at differences between the motor and the sensory neuron, you can see that this motor neuron has many dendrites, the sensory neuron has one dendrite. We can see that the cell body is at the end of the neuron, and here we can see the cell bodies in the center. Okay, these are just some differences as it was asked in one of the papers as well. Let's go to question 2.2. The diagram below represents the structure of a human eye. Now I've already prepared by coloring in the different parts of the eye so you don't get confused. 2.2.1. Identify part C. Now let's look at part C. I colored in C in blue and you can see it's at the middle layer. And the middle layer is the layer you will always be asked. And that is the choroid. Give one function of part E. I've colored part E in yellow. Yellow and it's the part that's holding the lens in place. So that can only be, of course, the suspensory ligaments. It holds the lens in place. Here, the other answers is connects the lens to the ciliary body and plays a role in accommodation. So that's the other answers. It was just easy for me to remember holds the lens in place. So you can remember whichever one is easier for you. State why the clearest image will form when the light rays fall on part D. Now this is part D. It's that little indentation in the retina. We already know that that is the yellow spot. And that's the place which has the highest amount or concentration of cones. So that is where the clearest image will form. Right, 2 to 4. Give one way in which part B is different from part F. Now, part B I've colored in is in red. It's the outer layer. That's the sclera. And part F is the lens. Now, we already know the lens is transparent. Why? To let light pass through. And B is the sclera. That's the white part of your eye. And the reason why it's white, it's because it's opaque. It doesn't allow light to pass through in all the parts of the eye. So there's it there. The sclera is opaque, doesn't allow light to pass through, while the lens is transparent, does allow light to pass through. So by the cornea and the lens only being transparent, light only enters in one part of the eye only. Right. That's a nice question. 2 to 5. Describe how the muscles in part A function. So part A, that is the iris. How the muscles in the iris function to increase the amount of light entering the eye. Now, amount of light entering the eye. So that can only be when it's dim light. So of course we know dim light, the pupils must dilate. So what happens? The circular muscle will relax and the radial muscles will contract. So make sure you also in your answer always have circular muscles of the iris, radial muscles of the iris contract and relax. 2 to 6. Describe how a blurred image forms if a person with normal vision wears spectacles with a biconvex lens while reading a book. Now this person has normal vision. You're wearing spectacles with a biconvex lens and you're reading a book. So your vision is normal. Your lens is going to refract light. You're wearing the spectacles with biconvex lenses. It's also going to refract light. And you're reading a book. So here, the image will obviously fall in front of the retina, and that's why the image is actually blurred, because it is near vision. Now, let's revise the eye. This is my diagram with the structure and the function of the eye. Structure and function of the eye. How the structure helps the function. That's known as adaptations. So, quickly, let's go through the eye. In yellow, I put the sclera. We know the sclera is the outer layer. It's a tough outer fibrous layer. And anything that's tough in the outer layer, it's there to protect the inner layers. In black is the choroid. It has melanin. That's why it appears black. It has melanin to prevent internal reflection of light in the eye. And the one that everybody forgets is the choroid has blood vessels to carry nutrients to the cells, carry nutrients and oxygen to the cells, and to remove carbon dioxide and waste from the cells. And then in reddish here, that is the inner layer, which is the retina. The retina has rods and cones. And that's the place where the image actually forms. We know rods are there for dim light and black and white vision, and cones are there for color vision and bright light vision. This little indentation here, that's the blind spot. There's no rods and no cones, so no image forms here. This other indentation in line with the lens, that's the yellow spot. That's the place where the clearest image forms, because this area only has cones. Then, this is the back of the eye. The back of the eye is filled with vitreous humor. It's a gel-like layer. It refracts light, maintains the shape of the eye, and distributes oxygen and nutrients to the back of the eye. This is the optic nerve, transmits impulses to the cerebrum. Right. Now, the sclera, the front of the sclera is convex. Anything that's convex will refract light, and the cornea is transparent. Anything that's transparent, like the windows, allow light to pass through. On the outside of the cornea is the conjunctiva. The conjunctiva has a lot of pain receptors, and that is for protecting the eye. This is the front of the eye. It's the anterior chamber filled with aqueous humor. The aqueous humor supplies the front of the eye with oxygen and nutrients, and also refracts light. This is the iris. It's the colored part of the eye, and the iris controls the size of the pupil. This is your ciliary body and your ciliary muscle, which holds the suspensory ligaments in place, and which in turn holds the lens in place. And these three structures, the ciliary muscle, the suspensory ligaments, and the lens, all play a role in accommodation. The lens is flexible for accommodation, transparent to let light pass through, and biconvex to refract light. Right, let me see if I've covered everything in this in the eye. And yes, I have. So this is a nice diagram to teach you how the structure helps the function, which is also known as adaptations. Right, let's look at 2.3. Endometriosis is a medical condition that occurs when the endometrium develops in or on other structures such as the fallopian tubes, ovaries, or pelvis. It's caused by higher than normal levels of estrogen. Females with this condition will most likely experience mild to severe menstrual pains. This condition can sometimes lead to infertility. Doctors may prescribe a contraceptive pill as treatment to reduce the development of the endometrium. This pill contains progesterone. 2.3.1. What structure? Name the structure where the endometrium normally develops. Now we know the endometrium is a part of the uterus. Explain why endometriosis in the fallopian tube may lead to infertility. So sometimes what happens is the fallopian tubes get blocked by the endometrium. It grows over the fallopian tube. So then the thickened layer of the endometrium blocks the fallopian tube, which will prevent the sperm from fusing with the ovum, which prevents fertilization. The other answer you can say is the thickened layer causes a blockage, prevents the embryo from reaching the uterus, so there's no implantation, which will lead to the death of the embryo or miscarriage. 2.3.3. Use a negative feedback mechanism to explain why pills containing progesterone are successful in treating endometriosis. This was a very smart question. Now we must know this. A high amount of progesterone will inhibit FSH. If there's no follicles at form, there'll be no estrogen that will be secreted, and that obviously will lead to less the thinning or less endometrium forming, as that is built up by the estrogen. Then 2.4, completely different from reproduction, the process of hearing. Now, here you must know the pinna of the ear traps sound waves. The auditory canal directs the sound waves to the tympanic membrane. The tympanic membrane vibrates, which causes the ossicles to vibrate. When the ossicles vibrate, these vibrations go to the oval window, which makes the perilymph vibrate, the endolum vibrate, then the organ of Corti is stimulated, and then the stimulus is converted to impulse, which is transmitted by the auditory nerve to the cerebrum. Now, I've made a very silly mnemonic. P, A, T, O, O, P, E, O. Pa2PO. Pa2PO, the P stands for pinna, the A stands for auditory canal, T for tympanic membrane, O for ossicles, the other O for oval window, then perilymph, then endolymph, and organ of Corti stimulated. That will help you with the pathway of hearing. This is a very good question. It has come out from 2014. Make sure you know this. It's now making its resurfacing again and appearing in more papers. Right, 2.5. The diagram below shows a developing human fetus. Now, again, I've taken preparations and I've colored in the blood vessels in beforehand. 2.5.1, identify part D. Now, part D is the membrane that's on the outer membrane in line with the chorion, and that with the chorionic villi, and that membrane is the chorion, because the chorionic villi grow from the or form from the chorion. 2.5.2, state two functions of the fluid in part C. The fluid in part C, it's the gray, and that's the amniotic fluid. Now we know the amniotic fluid acts as a shock absorber, protects the fetus from dehydration. It's made up of liquid, acts as a temperature regulator, and also helps with movement of the fetus. Now, 2.5.3, describe the development of the zygote until implantation. This is a question that comes out all the time. Make sure that you know this well. We know the zygote will divide by mitosis to form a ball of cells. What's that ball of cells called? The medulla, which develops into a hollow ball of cells called the blastocyst or blastula. That's very important. Make sure you know 2.5.3 for any exam. 254, state 2 ways in which Part A functions in protecting the developing fetus. Now, Part A. Part A is actually the whole part, the chorionic villi, and we know that's made up of the mother's blood vessels and the fetal blood vessels, so it's the function of the placenta. I would have said it acts as a filter against pathogens. I would have said provides antibodies. Here, the memo also says maintains endometrium and removes harmful metabolic waste. But the examiner and the marker will only mark the first two. So make sure you know this is a very good question. 255. Identify blood vessel B. Blood vessel B is the blood vessel that's taking blood to the fetus, so it can only be the umbilical vein. Make sure you know this. It's the vein. It's different from our blood circulation. 256. Describe how the nutrition of a human fetus differs from that of an oviparous organism. We know, the humans, the fetus receives nutrients from the mother's blood via the placenta and or via the umbilical vein. In oviparous organisms, the developing embryo receives nutrients from the yolk. Right, let's quickly revise the structure of the placenta. So the placenta is made up of the mother's blood vessels, fetal blood vessels, and then there's a sinus between the two, a space between the two. Then connecting the fetus to the placenta is the umbilical cord. The umbilical cord has two arteries, which is the umbilical arteries, and one umbilical vein. And the vein will carry oxygenated blood, and the artery will carry deoxygenated blood. Now, what will be in this blood? Don't ever say just oxygenated blood. What substances? Oxygen, amino acids, glucose, vitamins, minerals, antibodies. Deoxygenated blood, carbon dioxide, nitrogenous waste, urea, and uric acid. Right. I hope you enjoyed and you learned a lot from the question 3 from the November 2021 paper 1. And I hope you learned a lot in this video. Join me and watch all my other videos as well.